Okay, so topic 4.6, the cell cycle. Um, Next thing we need to talk about is how do our cells know when to divide, and how do they go through this whole process? Obviously, we know it's going to deal with our friends, the ligands, receptors, and some kind of signal transduction pathway, right? This should be something that you're expecting, right? This is how all of our cells communicate. Um, So a multicellular organism needs to coordinate cell division across multiple tissues and organs. This is true for us. Um, we need it for growth, right? If you weren't able to divide, you wouldn't have the trillions of cells you have as a human being. Um, we need to coordinate the timing of the cell division. Cells need to know when to divide. So up here I have some muscle cells. This is a slide with some muscle cells. And muscle cells don't really divide. They have no need to divide. They're very specialized, which means they have a very specialized look. So their cell division, if they were able to, it would look very strange because these are very elongated cells. They don't have this kind of normal cell structure that you're used to seeing. Um, these down here, these are layers of skin of the epidermis, right? You're going to see a lot of division here. You see a lot of division in these cells. Um, because these cells are constantly shedding, right? All of these layers up here are dead. They constantly need to be replenished. So they need to divide at a certain rate. Uh, Not all cells have the same cell cycle, right? Some cells, like such as skin cells, they're going to divide fairly regularly, and muscle cells will never divide. Muscle cells and nerve cells, neurons will never divide. Um, so some cells can trigger into a cell cycle. Um, some never trigger into the later phases of the cell cycle we're going to talk about. Again, things like muscle cells and nervous cells. But some are going to be kind of constantly dividing. You'll also notice types of cancer, since we'll be talking about cancer a little bit later. You're going to see cancer in cells that do divide, right? You won't really see cancer in cells that don't do a lot of division. Um, so it's just an interesting note for the future when we get to talking about that. So first, let's talk about our cell cycle right here. You have something called interphase. Interphase, it's labeled in blue around here. Interphase consists of G1, S, and G2. I'm going to write this in a more organized fashion up here. Interphase, G1, S, and G2. Interphase, there's also a G0, which we'll talk about in a bit. Um, but G1 is typically thought of as our growth phase. It's a cell doing what it would normally do, and it might remain at something called G0. G0 is a phase in which the cell is just operating normally. It has no plans to divide. It's not really growing. It's just doing its job. If the cell does begin growing, it might have received a trigger from some cyclins and CDKs, which we will talk about in a little bit. Don't worry. But basically, if the cell does begin growing and start getting larger, that means it's preparing for division. After G1, this phase right here, we move on to S phase. During S phase, this is the DNA synthesis phase. This might be something you remember from bio. This copies our chromosomes or replicates the DNA, however you want to think about it. Replicates the DNA, right? If you're making two cells, you need two sets of DNA. DNA replication occurs in S phase. Okay, the second phase, or the growth phase, G2, the second growth phase. This is when we are preparing for a process called mitosis. Mitosis, which is something you've heard of before, and we're going to talk about in another slide. Um, your cells begin growing more. You start to replicate all those organelles and proteins and stuff that you have in there that you're going to need. You start making a bunch of those. Your cell gets really big, and you get prepared for something called mitosis. Again, this is something you probably remember. But these are the three phases, the three typical phases of interphase, G1, S, and G2. Also, G0 is considered interphase. Obviously, something like a muscle cell would spend its life kind of in G0. Muscle or neuron, something like that, will spend its entire life in G0, will never divide. Something like a skin cell, you are going to see this phase, right? You are going to see this division. So let's talk about this next process right here, M phase or mitosis. Mitosis. talked about this a little bit in class. But why do you think we need to divide? What are some reasons to divide? You might think of something like asexual reproduction, reproduction without an exchange of genetic information. What about growth and repair? Growth and repair. These are all reasons that a cell would need to divide. So let's talk about how it looks. If you see the DNA in here looking like spaghetti, like it's all unwrapped, it's not condensed into chromosome form, you're probably looking at something called interphase, right? Again, this consists of G1, S, and G2. Of course, during S phase, you have replication of the DNA. So DNA might divide here. Then you have these, the nuclear envelope starts to disappear. You have these little spindle fibers start to form. You have centromeres kind of getting together. We've talked about all this stuff a long time ago. You might need to revisit some of your notes from when we talked about the cell. But what you start to see is this nuclear envelope or this nuclear membrane dissolve. You start to see these chromosomes begin to condense into these X-like structures. Of course, they look like that because it's replicated DNA. So this is DNA copy 1 of a chromosome. This is DNA copy 2. So this is replicated DNA, something to keep in mind. That happens during prophase. Prometaphase I never really get into, but it's a phase between prophase and metaphase, but let's just ignore that for now. I'm going to cross this one out because I don't think it's hugely important to know about prometaphase. Metaphase, all of your chromosomes, these spindle fibers kind of shake back and forth, and they kind of slowly move all of these chromosomes to the center of the cell, to this kind of metaphase plate right here. And they sit here to prepare for division, and then during anaphase, they slowly get pulled apart. So you're going to see each of these sister chromatids pulled to opposite poles of the cell. Let me label this. This is sister chromatids. This would be like 1 and 2, right? These would each be identical. 1 would be identical to 2 in this case, and they would be pulled to opposite poles of the cell. So ultimately, you end up with this process kind of finishing up with telophase. You end up with the nucleus reforming in telophase. You end up with these chromosomes kind of uncondensing and relaxing, unwinding. And after mitosis, you have something called cytokinesis. That is the phase in which the cell is divided into two. The cytoplasm itself is actually divided right here, and you have two cells. Two cells after cytokinesis. After telophase, you have two nuclei. So this is the basic overview of mitosis. I'll talk about some other stuff in class about how to remember these phases and what they do. Some of you might have heard of I peed on the mat C. I don't know if you guys heard that from your freshman bio class, or please make another touchdown. We'll talk about those in class. But this is a basic overview of mitosis. So from this video, what I would like you to remember interphase, the stuff that happens during interphase. Know where DNA synthesis occurs. Know that there's two separate growth phases on either side of this synthesis. And then understand the basics of mitosis and what happens. And again, we'll talk about some of this stuff more in depth in class, but the main thing you need to see is the big picture of this process. Okay, that's it for this video. Sorry, this one was a bit long.